poster that my colleagues and I are presenting at this meeting examines the issue of how you define the dose of a toxicant in a system where the exposure concentration, say in exposure water, changes over time. And in aquatic toxicology, we often assume that the external exposure dose is really, or exposure concentration rather, is the taken as the dose that the organism is actually exposed to at the site of action. And this isn't always the case when you have a system where, say, the concentration of a toxicant in water is declining rapidly, and then you have to think about the toxicokinetics, in other words, the rate of uptake and release. And so, this is my Ross Perot moment here. If you have, this is a figure from the poster, and basically the concentration outside the test organism, if it's constant throughout the course of the experiment, then you can have a steady state achieved, and the external exposure concentration is in fact proportional to the dose, which is the concentration inside the organism where all the bad things happen. However, if this is changing with time, then the kinetics become important, and unless you measure tissue concentrations or the dose, you may not know what this number really is, and therefore you have no idea, what is what the actual dose, and therefore what the lethal concentration is that you're of whatever response you're measuring. In our poster, we're start out by modeling the relationship between exposure and dose for systems where the external concentration is declining, changing with time, and ask the question, what's the appropriate exposure metric? What's the, how do you define the dose in a situation like that? Is it external concentration, initial concentration in the water, or is it concentration in the tissue? Secondly, we wanted to know from our modeling what the variables are that govern sort of uptake and release. And then finally, we did some case studies. And one case study involved a constant exposure scenario and looked at the things that govern the exposure concentration and dose inside the organism for that, and another one where there was definitely a declining dose scenario. Now, what we found with our modeling was that when you look at the rate of uptake and excretion, sort of this sort of uptake and release between external water exposure and the tissue concentration or the dose, where you're changing the rate at which your dose is declining. You can use a kinetic simple model to look at that, and where you have rate of uptake, a rate of release, and where you have these different curves are for different rates of decay. So if you're have zero constant water concentration, your dose, your tissue concentration reaches a steady state, which is this curve here. For different rates of decline in water concentration, you have different peak concentrations, and they occur at different times in the tissue. And this is very important, because what it means is that, especially if you're looking at systems like developing embryos, then at what time your peak concentration in the tissue occurs is going to be very, very important, because it's going to affect different stages of development. And so our paper exercise said, in a declining external concentration scenario, pharmacokinetics, rate of uptake and release, very important. You have to know that. And knowing, directly or indirectly what the dose is very, very important. And so in our first case study, we looked at data from a study done by workers where they exposed rainbow trout eggs and larvae to dieldrin and in a static system. And so that the only way you can change the concentration in a static system is by decline due to the organism absorbing it from the water. And so as if you absorb it quickly, water concentration goes down quickly. If you absorb it slowly, the water concentration goes down slowly as the tissue picks it up. Also very important in a static system is what the mass of your test organism total is, how many eggs or larvae you have, and the volume that they're in. Because the greater mass to volume, the more things are going to change because there's more absorbing going on. And this first study, these workers actually did a good job measuring using radio-labeled dieldrin uptake and release. They did a series of experiments where they measured the toxicology, and they were able to define a tissue concentration that had meaning in terms of an external dose that caused an adverse effect. The second case study we did, described on the poster, is a study done by Carls et al., where they measured the exposure of PAH, polycyclic aromatic hydrocarbons, obtained by passing seawater through rock columns that were coated with oil. And so as the water passed through the columns, oil partitioned, the fractions of petroleum, crude oil partitioned into the water. That was led into exposure chambers where herring eggs were sitting, and that's how they generated their exposure medium. The only problem with that was that the concentrations declined very rapidly, number one. Number two, they did two concurrent experiments where they let these columns lie fallow for a period of time, then restarted them, and then exposed a second batch of eggs where they called it a more weathered oil because it had been sitting on the column for a longer period of time. And they expressed their dose metric as total PAH, the sum of all the PAH analytes in water, and concluded that a 1 part per billion concentration of PAH was lethal, or it caused adverse effects, and that a more weathered oil was more toxic than less weathered oil. And I think the study showed a variety of problems because each analyte in a mixture, PAH, sort of 2-ring PAH, 3-ring PAH, 4-ring PAH, and their alkyl homologs all had different pharmacokinetics, number one. So they declined in concentration at different rates. Their uptake by the eggs occurred at different rates. And as a result, at no point in the experiment did they really know what the true concentration at the site of action was, or what the peak concentration. They did measure tissue concentrations, but the problem was that for many of the experiments, they didn't measure them soon enough because of the rapid changes that were occurring in the water. And so at the end of the day, one isn't really sure what the concentrations at the site of action, in other words, taken as tissue concentrations, were that caused the effects that they observed. And so the take-home from this poster and the study is that if you have a system of where your exposure concentrations are changing rapidly, you have to be very careful about how you define dose, and using some initial water concentration or some average water concentration isn't going to be accurate because of the changes that occur in the dosing medium with time, especially if it's a mixture, as in the case of the crude oil study. far better to use peak tissue concentration, which you have to measure. But in order to do that, you have to design your experiment so that you make your measurements quick at sufficiently close time intervals so that you don't miss that magic moment where you have your peak concentration. And finally, again, in dealing with mixtures, you really need to know what the sort of pharmacokinetics are and the toxicological properties, the potency of each of the components in that mixture in order to interpret your data properly. And so that's kind of the, what the poster's all about. In other words, dose, makes the poison, not exposure.